If this is your first time here, subscribe now. Because what you're about to learn, most people never will. A laboratory in northern Italy ran genetic sequencing on a bloodstain older than the country itself. What came back wasn't contamination, it wasn't dye. It was a full biological profile, belonging to a man, on a cloth with no birth record, no family tree, no living relative to compare it against. A geneticist at a major university confirmed the sample was human, male, and centuries old. Then came the part nobody could explain away. Every Y chromosome in nature comes from a father. This one has no father on file. No sibling. No cousin 3 generations removed. Just a chromosome sitting inside a 14-foot piece of linen with absolutely nothing to check it against. That's not a footnote, that's the entire case, in one sentence. The most tested religious artifact in human history cannot be compared to a single verified sample of the man it's supposedly from, because no historic DNA profile of that individual exists anywhere on Earth. Not in a museum, not in an archive, not in a private collection. It simply isn't there. So when a lab says it confirms something about this cloth, the obvious question almost nobody asks out loud is this. If there's no father on record, no comparison DNA anywhere on Earth, what exactly did the lab just confirm? Start with what almost everyone already agrees on. The Shroud of Turin is a 14-foot linen cloth bearing the faint image of a crucified man, front and back, head to head, as if a body had been laid on one half and the other half folded over the top. It has been housed in Turin Cathedral since 1578, brought there by the House of Savoy, and displayed to the public only a handful of times per century. Church tradition connects it to the burial cloth described in the Gospels, wrapped around the body after crucifixion. That's the origin story most people have heard. It's simple, it's old, and for decades it looked settled. In 1988, 3 independent laboratories, Oxford, Zurich, and Arizona, carbon-dated a small sample cut from the cloth. Their combined result placed the linen's origin between 1260 and 1390 AD, medieval, not 1st century. Not close. For the broader scientific community, that single test did what centuries of theological debate couldn't. It closed the book. Case dismissed. Likely a devotional artwork, possibly a deliberate forgery, created roughly 13 centuries after the events it claimed to depict. But 10 years before that dating test, a different kind of investigation had already complicated the picture. In 1978, a team of scientists calling themselves the Shroud of Turin Research Project, STURP, were given unprecedented direct access to the cloth. They spent 120 hours running the kind of forensic battery you'd expect in a modern crime lab: spectral analysis, microscopy, chemical testing. Their conclusion wasn't forgery confirmed, it was stranger than that. They couldn't identify the substance forming the image, couldn't replicate it with period-appropriate pigments, and couldn't explain how a medieval artist, if that's what this was, had produced an effect no known 14th century technique could produce. The debate shifted. Not is this real, but how was this image even made? For years afterward, the 1988 carbon date stood as the trump card. Medieval cloth, unexplained image, mystery contained. Case closed, until it wasn't. Because starting in the 2010s, new chemistry and new genetic testing began pulling at threads the carbon date never accounted for. And what they found didn't confirm the medieval story. It undercut it. Here is where it gets complicated. In the past decade, a materials scientist named Dr. Liberato De Caro, working at Italy's Institute of Crystallography, applied a different dating method entirely: X-ray analysis of the linen's structural degradation, rather than carbon isotopes. His results didn't land anywhere near 160 to 1390. They pointed toward roughly 2,000 years old. Not medieval, not close to medieval. A gap of over 1,000 years between two supposedly reliable scientific methods, both testing the same object. That alone would be enough to reopen the case. But the genetic side of the story is where it gets stranger. A professor of genetics and genomics at the University of Padua, Gianni Barcaccia, led an analysis of DNA trace material recovered from the cloth's fibers. This wasn't a simple yes or no test. Think of it less like a single fingerprint and more like reading footprints layered on top of each other in wet sand for 2,000 years. Every hand that ever touched the cloth left something behind, and the lab had to separate hundreds of overlapping trails from one another. What Barcaccia's team found, published in a study years earlier, was a geographic signature: DNA fragments consistent with more than 55% Near Eastern origin, close to 39% Indian origin, and under 6% European origin. Sit with those numbers for a second. A cloth housed in Europe since at least the 1300s, tested by European labs, in a European cathedral, and the majority of the genetic material recovered doesn't trace back to Europe at all. It traces to the Near East and to South Asia. That single finding undoes the tidy medieval forgery narrative from a few minutes ago. If this cloth were manufactured in 14th century Europe as a devotional prop, the genetic footprint left behind by everyone who touched, wove, or handled it over the centuries should skew overwhelmingly European. It doesn't. It skews east, heavily east. Two different scientific methods, X-ray dating and DNA geography, now point in the same unexpected direction, and neither one agrees with the carbon date that supposedly closed this case in 1988. Adding one more name to the file, Professor Pierluigi Baima Bologna, a forensic medicine specialist, identified human blood of type AB on the cloth back in the 1980s, long before modern DNA sequencing existed. It was Baima Bologna's own preserved samples, kept for decades, that fed directly into the newer genetic studies. He didn't live to see the follow-up research published, but the material trail runs directly from his original forensic work to Barcaccia's genetic lab, decades later. Clean chain of evidence, except for what it actually proves. Because confirming a 2,000-year-old Near Eastern heavy geographic history for this cloth is one kind of finding. Confirming whose blood is actually on it, that's an entirely different problem, and it's the one nobody's DNA test has come close to solving. The first theory says, none of this is as significant as it sounds, and it comes from outside the genetics field entirely. René Enevold, a geoscientist at the Mosgaard Museum in Denmark who has spent years analyzing ancient pollen, has raised direct challenges to how the shroud's biological trace evidence gets interpreted. Her core criticism, plant DNA recovered from an object like this could have come from dozens of possible sources over centuries of handling, travel, and storage, and there's no reliable way to trace a single grain back to one specific origin point. She's also pointed out a more technical problem. The genetic identification in these studies often only reaches the subgenus level, a broad category, not the species level that would actually be needed to pin down a precise geographic origin. In plain terms, the tests can tell you, this came from a plant family found across a wide region, not this exact plant grew in exactly this exact place. Researchers are careful to frame conclusions like the 55% Near Eastern, 39% Indian breakdown as probability and pattern, not certainty. Even Barcaccia himself, when pressed on what the DNA geography actually proves about the cloth's ultimate origin, was blunt. We cannot say anything more on its origin. That's the ceiling of Theory 1. Every hand that ever touched this cloth, pilgrims carrying it through Near Eastern trade routes, handlers transporting Indian-made linen for religious use, restorers working on it across centuries in multiple countries, could produce exactly this genetic signature. Contamination, traffic, coincidence stacked on coincidence. Under this theory, the DNA proves people moved. It doesn't prove who was wrapped in the cloth to begin with, which would be a tidy way to close this file, if it weren't for one detail Theory 1 can't fully explain. That 39% Indian genetic signature isn't just random noise from anonymous travelers passing through anonymous ports. There's a far more specific, far more contested explanation for exactly where that Indian DNA might have come from. And it goes back not to trade routes at all, but to a single building in Jerusalem, and a single day on the religious calendar. A single building in Jerusalem. A single day on the calendar. That's where the second theory starts, and it doesn't treat the Indian genetic signature as an accident at all. The second theory points to something far more specific than pilgrims wandering through ancient trade routes. Historical and textile records describe a particular fabric used in the temple in Jerusalem, fine linen imported from India, reserved for the high priest's garments during the rituals of Yom Kippur, the Day of Atonement. Not everyday cloth, ceremonial cloth, sourced from a specific region, for a specific religious function, at a specific point in the 1st century. Run that against the DNA numbers again. Roughly 55% Near Eastern genetic material, close to 39% Indian, under 6% European. Under the first theory, those numbers were just noise. Centuries of random hands leaving random traces. Under this theory, they're not noise at all. They're a signature. A cloth carrying Indian genetic material at that concentration, recovered in a context tied to Jerusalem, lines up specifically with a documented 1st century temple practice, not with generic contamination from anonymous travelers over 1,000 years. And that's the problem for Theory 1. If the linen's origin traces to a specific ceremonial supply chain rather than random contact over centuries, just people passing through stops being a sufficient explanation. The geography is too precise, two aligned with one particular practice, on one particular religious calendar, in one particular building. That doesn't prove the theory, it just means the tidy dismissal from a few minutes ago no longer covers all the evidence. Here's the limitation, stated plainly, because researchers are careful to frame this as probability, not certainty. A shared origin between the fabric and a known historical practice is not the same as proof of who was eventually wrapped in it. Linen from the right place, used in the right kind of ritual, could still end up crossing paths with the cloth for reasons that have nothing to do with the crucifixion. Correlation, not identity, which brings us to the third theory, the one most researchers are hesitant to say out loud, because it doesn't sit in the safe, hedged, we cannot say anything more territory, the way the first two do. This one goes back to the anomaly that opened the whole case. The Y chromosome. The one with no father on record, no comparison sample, nothing to check it against anywhere on Earth. A geneticist named Shroud of Turin, who studied the shroud's biological evidence directly, came away from that research with an intense personal conviction that the cloth is, in fact, the burial cloth of Jesus Christ. Not a hedge, not a probability, a conviction from a trained geneticist built on the same DNA data everyone else in this story has been treating as inconclusive. But here's the counterweight, and it's not optional. The forensic DNA literature on this exact question is blunt, almost cold about it. DNA sequencing cannot be used to determine the identity of the man in this cloth because no historic genetic profile of that specific individual exists for comparison. Anywhere. Ever. There is no reference sample. There never was one. Which means for Turin's conviction and the forensic community's caution aren't actually contradicting the data. They're both standing on the exact same missing piece of evidence and reading the empty space differently. One side sees faith confirmed by absence. The other sees a scientific question that cannot, structurally, be answered. Nobody outside either camp gets to declare a winner here. Not yet. Picture the lab where the most recent chapter of this got written. Padua, Italy. A controlled sequencing facility. Sometime in early 2026. A research team is working through material first collected back in 1978, during the original STIRP investigation, samples that have sat, preserved, for nearly 5 decades, waiting for technology to catch up to the questions being asked of them. The fibers going into the sequencer aren't clean. They can't be. 2,000 years of handling, storage, and exposure have left the genetic material fragmented, degraded, mixed. Every run through the machine isn't reading one clear signal. It's untangling dozens of overlapping ones, layered on top of each other like handwriting traced over and over on the same page, until the original strokes are barely visible underneath the additions. The team is looking for mitochondrial DNA, the genetic material passed down strictly through the maternal line, distinct from the nuclear DNA that carries a full genetic blueprint. Think of it as a much shorter, much more limited logbook, passed mother to child, generation after generation, largely unchanged. What comes back is a list of lineages. K1A1B1A, H2A2, H1B, H33, 4 separate genetic threads recovered from the same small sample. And one of them breaks the case wide open, for exactly the wrong reason. The lineage K1A1B1A matches the known mitochondrial profile of one of the original 1978 sample collectors. Not a first-century pilgrim, not a temple attendant, a 20th-century scientist who handled the cloth during the original investigation, whose own genetic material ended up inside the very sample being tested for ancient origins. Sit with that for a second. Modern contamination. Confirmed contamination. Not a hypothetical risk raised by a skeptic on the sidelines. A direct match, inside the data, to a person alive within living memory. Here's what that does to everything before it. Every geographic percentage. Every Near Eastern signature, every Indian lineage, every carefully footnoted number from the earlier theories. All of it was recovered using the same collection methods, handled by the same category of researchers, decades after the fact. If one lineage in this exact sample set traces cleanly to a 1978 scientist, there is no guarantee, none, that the rest of the genetic geography isn't also, at least partly, a portrait of 20th century hands, rather than first century blood. The honest position, the one researchers who work on this material actually hold, isn't solved. It's split. Genuinely, professionally split. Some read the Near Eastern and Indian signatures as residue of an ancient journey. Others read the same numbers as residue of modern veneration, modern tourism, modern handling, pilgrims and scientists and restorers, all leaving their own genetic fingerprints on top of whatever original signal might or might not still be underneath. Not proof of authenticity. Not proof of forgery either. One retired conservator who spent years working near this material put it simply, in an unguarded moment, to a colleague, "You can clean a fingerprint off glass, but you can never be completely sure you got all of it." That's the entire problem with this cloth, compressed into a sentence. Every test since 1978 has been trying to read a signal that keeps absorbing the noise of everyone who goes looking for it, which is the real shape of this story if you zoom out far enough. This isn't really about one bloodstain, or one chromosome, or one disputed percentage of Indian linen. It's about what happens when the most scientifically examined object in human history keeps meeting the absolute limit of what DNA can actually prove. Every new instrument, every new sequencing method, every new laboratory in Padua or anywhere else gets thrown at this cloth expecting a final answer. And every single time, the result comes back the same way. More detail, more geography, more named lineages, and still, at the center of it, an identity that no genetic test on Earth has the reference material to confirm. That's not a failure of science. It might be the most honest thing science has said about this object in decades. Some archives don't get fuller the more you dig into them. They get more layered. One page of handwriting laid over another, until the original ink and every later edition are woven into a single, inseparable text. And no lab technique yet built knows how to read only the first line. Scientists finally tested the DNA. The results didn't answer the question everyone wanted answered. They rewrote it into something even harder to close the book on than before.